Right now, dissolved in your blood, there is a molecule of glycine, the simplest amino acid there is. And that molecule is carrying two full electrical charges, a positive one on its nitrogen, a negative one on its oxygen. Two charges. And yet, if you measured its net charge, you'd read exactly 0. Here's the part that should bother you. That same glycine sitting at the pH of your bloodstream is almost useless at doing the one thing chemistry students assume amino acids are good at, resisting changes in pH. At the pH where your body actually lives, glycine is one of the worst buffers you could pick. Both of those facts come from one idea. An amino acid is not a single molecule. It's a shapeshifter. And today you're going to learn to predict exactly which form it takes and what charge it carries, at any pH you throw at it. Start here. Stop thinking of an amino acid as a molecule. Think of it as a family of related species that interconvert as the pH of the solution changes. Add acid and protons pile onto the molecule. Add base and protons strip off. Every ionizable group flips independently, and each flip changes the charge. The tool that tells you which form dominates is the Henderson-Hasselbalch equation. pH equals pKa plus the log of the deprotonated form over the protonated form. Don't memorize it yet. Read what it means. The pKa is simply the pH at which a given group is exactly half dissociated. Half the molecules have given up their proton, half are holding on. That single number is a fulcrum. When the pH sits below a group's pKa, the group holds its proton, it stays protonated. When the pH climbs above the pKa, the group lets go. It becomes deprotonated. At the pKa itself, it's a 50-50 split. So charge for an amino acid is just bookkeeping. Walk up the pH scale, and every time you cross a pKa, one group flips and the net charge steps down by 1. Now the single most common mistake, and I want to kill it right now. Students learn that the isoelectric point, the pi, the pH where net charge is 0, and they assume you get it by averaging all the pKa values together. That is wrong. The pi is the average of only the two pKa values that sit on either side of the neutral, zero-charge species. For a simple amino acid, those happen to be the only two you have. But the moment a side chain gets involved, you have to find the neutral form first, and then average the two pKa's that bracket it. Frame it that way, and you'll never miss a pi again. Let's build it with glycine one step at a time and watch the charge change. Glycine has two ionizable groups, an alpha carboxyl group, the COOH, with a pKa of about 2.3, and an alpha amino group, the NH3+, with a pKa of about 9.6. Drop glycine into strongly acidic solution, pH 1. That's below both pKa values, so both groups stay protonated. The carboxyl is COOH, neutral. The amino group is NH3+, positive. Net charge +1. Now slowly raise the pH. As you pass 2.3, the carboxyl group crosses its pKa and lets go of its proton. COOH becomes COO-, negative. Look at the molecule now, a negative carboxylate, a positive ammonium. Two charges, opposite signs, on the same small molecule. Net charge, 0. This form has a name, the zwitterion, from the German for hybrid ion. And here's the correction to the second great misconception. A zwitterion is not uncharged. It is intensely charged. It just carries equal and opposite charges that cancel in the net. At the pH of your body, essentially every free amino acid exists as a zwitterion. Net neutral and no charge are not the same sentence. Keep raising the pH. Nothing dramatic happens for a while until you approach 9.6. There, the ammonium group crosses its pKa and gives up its proton. NH3+ becomes NH2, now neutral. You're left with a negative carboxylate and a neutral amine, net charge, -1. So glycine climbs a staircase, +1 down to 0, down to -1, as pH rises. And somewhere on the way down, it passes through that zero-charge zwitterion, The pH where it sits exactly at net zero is the isoelectric point, the Pi. And using the rule from a moment ago, average the two pKa's flanking the neutral species. Glycine's Pi is 2.3 plus 9.6, divided by 2, about 5.97, just under 6. Now extend this to an amino acid with a charged side chain, say glutamate, which adds a second carboxyl group on its side chain with a pKa near 4.25. Now you have 3 pKa values, 3 flips, and charges running from +1 all the way to -2. To find its Pi, you don't average all 3. You find the species with 0 net charge. See that it's bracketed by the 2.2 and 4.25 carboxyl pKa's, and average those. Glutamate's Pi lands down near 3.2, acidic, exactly as the extra negative side chain demands. Here's the experiment that proves the whole framework: the titration curve. Take that acidic glycine and add base, drop by drop, plotting pH the whole way. You get a curve with 2 flat shelves, 2 buffering regions, one centered on each pKa at 2.3 and at 9.6. On those shelves, pH barely moves as you add base, because the half-protonated group is soaking up the change. That's buffering. But between the 2 shelves, right around the Pi near 6, the curve does something violent. It goes nearly vertical. A tiny splash of base sends the pH rocketing upward. At its own isoelectric point, glycine has almost 0 buffering capacity. Sit with that. The Pi of glycine, about 6, is close to the pH your cells operate at. And that is the exact pH where glycine is least able to resist a change in pH. The place it looks most neutral is the place it's most fragile. That's why life does not lean on amino-terminal groups to buffer the blood. So which groups does biology use? Follow the pKa. First, buffering inside proteins. The one side chain whose pKa sits right near physiological pH is histidine at about 6.0. That is not a coincidence. It is why histidine is the workhorse pH buffer of proteins and blood. It's the residue behind the Bohr effect, where hemoglobin picks up protons in acidic CO2-rich tissues and releases oxygen where it's needed. Histidine works precisely because it's half-protonated at the pH of the body. The same condition that made glycine a bad buffer makes histidine a great one. Second, separating proteins. Every protein is a bag of ionizable groups, so every protein has its own Pi. Techniques like ion exchange chromatography and isoelectric focusing exploit this directly. Run a protein through a pH gradient, and it stops migrating exactly at its Pi, where its net charge hits 0. And at that same Pi, a protein is at its least soluble. The charges that normally keep neighbors repelling each other have canceled, which is why proteins tend to precipitate at their isoelectric point. Third, the chemistry of enzymes. Inside an active site, a residue's pKa is not the textbook number. Bury a glutamate in a nonpolar pocket or park it next to another charge, and its pKa can shift by several units. Enzymes weaponize this. In the catalytic triad of serine proteases like chymotrypsin, a histidine's pKa is tuned by a neighboring aspartate so that it can grab and release a proton at exactly the right instant to cleave a peptide bond. The whole reaction hinges on one residue being poised at the edge of its protonation state. So remember this: an amino acid's charge isn't a fact you memorize. It's a story you can read straight off the pKa values, one proton at a time. Find the pH, count the flips, and the molecule tells you exactly who it is.